Okay, so let's move to the second part. Let me share the screen now. Okay, great. So we stopped here, and I would like to take forward from here. So what we understood, we have to have proper ethics in our mind. We should be properly trained. We should be alert. Uh, Still, we have the best people, best lab, best systems in place. Still, we have in some gaps. So we should be ready to learn new things. We should be ready to learn changes. We should be adaptable, right? Ultimately, we should be innovative enough to have solutions for all the challenges, upcoming challenges in our hand. We are definitely, as a green lab, we are targeting to have paperless environment, and ultimately my lab has to be fit for the purpose. So let's move towards one step closer to green lab. So let's move to the second part of this my talk lecture. So let's understanding the expectation of a green lab. So what I do, I'll compare based on my experience. I'll compare the labs operating in past and how they are operating in present, or maybe they are going to operate in future. So past, if I talk about method development part, we used to use notebook, a physical long or medium-sized long notebook. There we used to record each and everything, including solution preparations, experimentation results, inferences, references, everything. Then how to go ahead, planning for next day. So everything handwritten nicely with those books. Ultimately, the doer and supervisor used to sign also, ideally every day, daily basis. Sometimes used to get delayed, so we used to take sign for 2 days with the dates, actual dates, present date. So anyways, so what is going to be changed now? Let's understand. So first of all, how we used to do earlier in past, manually in notebooks, handwritten, and you can understand, including if I talk about my handwriting, it's difficult sometimes to even read myself. Okay, so it's not a good scenario, but we didn't have any choice that time. So everything started with handwritten in manual notebooks with properly designed notebooks, but handwritten. It was extremely tedious. Away from online, so many times it happens. It used to happen like I couldn't compile, if I take my example, the same day. When did I did experiments, I start writing, documenting, but that is night 8 o'clock. And then 9 o'clock, there was no mood. And then we used to think tomorrow we'll do before experimentation. So many times we are getting away from online. Inconsistency was a challenge because my style may be different from others. Compilation may be different. Lengthy, shorter, depends on individual whim and wish. Not suitable for regulated job, I think, because of inconsistency and own styling. CRO managed with extra efforts. So that time I was working with CRO, we did with extra efforts. But then consistency and training was not easy, especially to get consistent outcome, because manual intervention was very significant. When I talk about today, how we can operate, so the handwritten thing can completely be replaced with e-notebook in automation, a kind of automation. Like there, I need to write not even 10%. Maybe I need to do signature digitally. I can sign it off. Even there, I don't need any handwritten signature. So that is available, those kind of solutions available. So with e-notebook, what I used to do handwriting for most of the experiments and presentation and documentation and presenting to my supervisor, which can be 100% converted into, replaced with the e-notebook into automation. Now, another thing is handwritten was extremely tedious. It is quite manageable. Time is good. You can manage your time nicely on time, because we are doing, and like we have tablet or computer, like we can simply say drag and drop kind of features. Like we have multiple options: sodium acetate solution, ammonium acetate solution. What kind of material you are taking? Ammonium acetate, just drag, select, drag. You drag, it will be like coming as a step a third of that particular solution preparation. That's fine. It was difficult to regulate. Here it is easy to regulate, and other options are also like higher options available. It is very suitable for regulated job, regulated environment, regulated bioanalysis. Now, what used to happen, I used to document, and as I mentioned, for a bioanalyst, we have to have, we used to go give 50% time in documentation earlier, my days, my career's day. But now, that extra effort, because I'm writing by hand and my writing was bad and slow, both. So you can understand how much extra efforts. But then today, if I work with the same lab and with the new system, with the e-notebook, I really get at least 20%, 25% time in my hand, which can be utilized for science. So extra efforts, if I'm putting, even the remaining efforts I'm putting, it will be for science, which is definitely having more value addition, more value for money. Consistency and training was a challenge, but I think were challenges, but now consistency and training quite manageable with e-notebook. So I'm now moving towards, you can understand, paperless, as I mentioned. So present and past and present. So earlier, when I talk about chromatographic outputs, chromatograms, vendor-driven formats, they used to give fixed formats, like this will be the report, this will be the chromatogram, here we have title, this and that. You cannot change. You didn't have option. But now with the application of, with the introduction of LIMS, so here you can understand, I am using short forms of laboratory informatics management system. This is LIMS, and then SDMS, scientific data management system. So if I'm talking about here in this case, where we have fixed formats coming from the vendors, with the help of LIMS, you have options for formats, especially for bioanalytical lab. Now, customization was impossible. Nobody was trained, engineers as well. When I talk about 1998, 2000, when I started my career, and Shimadzu, I used to operate as HPLC, and they had very good software as per that updated. And they had inbuilt feature for calibration curve with drawing and everything. But when I talk about the formatting of chromatogram, which was coming out of that machine as a raw data, which should be actually intact. But if I want to do some change, even the right things like date format as per my SOP, it was difficult. Even engineers were not trained on their application part, so it was very difficult to do that. But today, with the implementation of, with the advancement of science and their development in software side, and with our introduction of LIMS and everything, customization is very easy. But this customization will be reflected through audit trail. So whatever I'm doing, it should be through password protected, through passwords, through my login. So everything will go. Manish has done this report, this date format change from this to that. Everything will be recorded. So it is 100% 21 CFR Part 11 compliance system, which is being required, but customization is possible with the regulation. So it was extremely tedious earlier. Now it is manageable, as you understand. Time consuming, now in time. You can do very quickly because things are available, resources are available, and application is like that, supporting. Not suitable for regulated. Earlier it was very difficult because if my SOP says date format is 10th March 2020, and it is giving 10 MAR apostrophe 20, then it is a difficult thing. So my SOP deviation, or I need to change the SOP, which is again a tedious work. So sometimes that time we were, as I mentioned, extra work. So we used to do these kind of things. We used to get the software validation, this and that, machine installed, and before that we made the SOP, then we went for revision. That we used to do a lot because even the updation used to come 6 months, every 6 months. So that is a job, a lot of work. Overall, the consistency and training were not easy in that scenario. Here, consistency and training easy because it is highly customized and suitable for regulated. Next is regarding calibration curve. So earlier, even before when I started my career, so many other labs except of our labs, we got Shimadzu advanced system, Shimadzu HPLC advanced system. So there I used to do lot of analysis, bioanalysis using HPLC. And they had automated feature for constructing calibration curve with Smartji software. But I have seen, I discussed, interacted with so many my peers in that point of time, in that era. They didn't have this. So they used to use Excel sheets. Most of the labs, I was a part of luckily advanced lab named as Lambda Therapeutic at Ahmedabad. Probably they were named as conducting first GCP compliant bio study in India. The team came from Ranbaxy. Ranbaxy was very big. They only taught us, and then we created. So that is what history. But then, except of that place, I find so many people, they were using Excel sheet to calculate, draw those calibration curve, lot of equations and this and that, and then they feed in Excel. Then they were getting subparts and then final results. Very tedious process. Okay, even then they design, you need to put lot of input you have to give in Excel sheet. Then only you get the final results, and then no validation of those Excel sheets. So it will not be reliable. Anyways, so, but this software came to us, it was fully compliant. There we used to construct calibration curve through software. So we become very efficient. We used to analyze, in place of people used to analyze only 50, 60, 70 samples, maximum 90 samples. We used to analyze 200 samples in a day. But that was the part, because a lot of time we used to invest in creating calibration curve, then calculating everything in Excel, then used to report. So the post-analysis phase was very tough. Reporting part was very tough and time-consuming, tedious for us. But with the introduction of this simple software updation, it was fantastic. And then later on in some advanced version of laboratories, when we develop, we find even LIMS in place. So LIMS will take all concentration value, not even software required, take concentration value from the machine, and then they do their all integration, construction of calibration curve, calculation of quantitation of all values they will do automatically. So this is a further automated, further efficient, and we save a lot of time. To and fro, manual intervention, a lot of time we can save. And in place of manpower, in place of 5 people or 3 people working for 3-4 hours, you can do this job within half an hour maximum with these software solutions in today's era. So it's where it has become very efficient, which is again a part of advancement. But based on industry experience, even our software engineers feedback, scientist feedback, they developed, they also advanced, they learned a lot, even suppliers. Okay, next is, it was like when you say using of Word copies, Excel sheets, software, machine, it's an open network, lot of interventions, so a lot of margins of errors. But when you have integrated system like LIMS and even the inbuilt software capabilities for calibration curve construction, you have excellent controls, less interventions, manual interventions, so less error. Yeah, so it's a better system. No IT, software, validation. That time validation was not at all a concept, especially software side. We need to validate even Excel sheets that I learned later stage of my career. So today, everything and anything, whatever you're using, even software, small applications, you need to validate. Only then it is, say, compliant and you can present your data. Otherwise, you'll be in big trouble. So IT software validation now, whatever applications, softwares, or we are getting fully supported. So they are fully validated. Manual interventions, as I mentioned earlier, a lot of different interventions because of different applications we were using. So a lot of manual interventions. Now, less manual intervention or almost no. Repeated errors, a lot of errors used to happen even after QC and QA. Concentration values changed here and there, one value, 1 point something, lot of, then even integration, like you are having decimal places. Software having algorithm different 16 digit, this is having, computer is having 24 digits, so you always get in points, second, third digit, you get changed value, and that is a big challenge. No integration. But now everything is integrated. So not those kind of errors happening. Time consuming, yes, now very quick. Key resources wasted for quality control. Now, even if we do a lot of hard work and compiled and recompile, rechecking, lot of resources used to be wasted for that, those kind of activities, which were not right now. If you think of with the introduction of softwares, advanced softwares and LIMS, you can save a lot of resources which can be utilized for science and value creation, which will be having higher value. Not at all regulated. Today, everything is regulated, quite suitable, right? And again, training and through that consistency is quite manageable with all these new solutions. Earlier, which was a challenge. Coming to the data movement, data transfer from lab to other departments, other places, like instrument to works, Excel sheet, and then PK software, because we need to do PK analysis, whatever we are generating data. It's a part of clinical research. So we do a lot of pharmacokinetic calculations. And then finally, we propose whether study is pass or fail. If a study is going good, passing, then we submit the study to the sponsor, and then they submit to the regulatory agency. So a lot of data movement. Like if concentration is getting generated on equipment, you understand, till reaching to the final report, B report, even it will reach to PK software, then we'll have bioanalytical report, then bioanalytical report will be a part of statistical report, bioanalytical report, clinical report is a part of B bioequivalence report. That will go. So many times you need to transcribe, and everywhere you have different tables. There you need to feed these values. So even computer to computer, computer to machine, algorithm is changing. So decimal places may change. And then it is sometimes if you miss, your QC is miss, the wrong value goes off, which is a miss, again, non-compliance. So it was a big challenge, data transfer, data movement in lab, and in even the bioequivalence centers, research centers, because of open network. Lot of interventions. Today, excellent control, fully automated, no process of validation, but today as per regulatory, you need to validate everything, which is possible with these solutions. No manual intervention, less or no. Today, if I talk about errors, a lot of errors used to happen because I was part, I have seen that. Today, it is nothing, almost nothing. We were very cleanly submitting these reports, big reports without any error, without finding. So many cases I had in my career where we cleared, these products are now in the market also, US market, Europe market, without any findings. Key resources, as I mentioned, wasted for QC and other jobs. Earlier, now you can use for science, so we can do better science. My knowledge can be utilized for science, not for QC, just checking the document. My time is important. My knowledge can be used for better, not only mine, analysts and other level people. Okay, so very suitable for regulated bioanalysis, even for training. Now, another important aspect is reporting. So report generation was a big challenge. Now with the application of advanced features, softwares, and even the machines, directly concentration values are going to different report formats which are fixed, especially through LIMS and even through the machine. So even you have now simple solution, you don't have LIMS, your lab is quite compliant. But with the LIMS, I'll give you an example how efficient people will come. Obviously, it is having value. It means you need to invest money for that. But then it is really value for money. That money can be recovered in a year's time maximum, because you have a lot of savings, lot of quality proven savings, time, money, resources, and then consistently, if you are providing quality data, then you get repeated business. If you do errors, if you do mistakes, that client will not come, once it goes away from you. So good quality will ensure the repeated analysis and business for your lab also. So it is in a longer run, it is very important. So when I say how this report generation, so it is fully automated, okay, fully automated. And I say when initially you introduce this system like LIMS and SDMS, SDMS is basically for data archiving. So you collect everything, report, data, everything you transfer and put, like physical archive. We have archiving facility there. We fill all the racks, we fill put all the files. Same way, all the data folders, everything will be go to centralized server. There you have all the data. So retrieval will be like on tips. You do metadata, you do lab informatics, everything will come. You do simvastatin, all the files related to that data, everything will come. So it is very good and easy. Okay. So I say sometimes when you introduce something new like lab informatics system or scientific data management system, it may not be familiar to the worker, like the doers. They say, no, sir, we are very good in existing system. Why you are introducing new? But we as a management understand where the world is going. Now the regulatory or agencies, earlier they were not asking anything, but now they are asking electronic formats because they know if you don't have manual intervention, then their audit time will also be reduced. They will do their job quickly. Otherwise, they need to check so many things. So it is a world and it's a change, which is better for all, for them. So when you introduce this system, which are efficient, and as per current system, efficiently, we can do the job, not only doing the job efficiently, these systems, whatever systems we are having in the lab will become inspector friendly. Doers may have some challenge in the beginning. After that, they also be happy. No, sir, abhi to error nahi hota, and we are very happy. See, earlier I used to have 3, 4, 5 findings in my study, but now no findings. We are doing very good. They will be happy because we know it is going to happen. Earlier, first day, new change. Change is always painful. It is painful unless your vision is not clear. If your vision is clear, it is okay. You can accept. Because then you start getting the good results and you start feeling less pain, more pleasure. And that is what the way we go ahead. So it's more happiness. So these things are inspector auditor friendly. Next is archiving. So archiving is a big activity. Archivists, as per GLP, directly report to the management, because that nobody should have say on the data. So earlier, when I see or reminded my career, archiving area was used to look like a dump yard of the lab or company. No indexing will make our life hectic. If somebody does mistake in indexing, if you are not able to prepare your titles properly, indexing, gone. It is very difficult to find that data then later. Indexing for racks, indexing for files, everything. So poor system for that will make used to make our life very hectic. So today, if I talk about, because of introduction of especially scientific data management, SDMS is highly integrated and most important area. Why this is most important area? Not only to beautify, it is the most important area for auditors, because they have only one thing by which they can see whether a study is being conducted or conducted then properly conducted, and they have only one way to have proper documentation and documents available with you, by which they can claim, they can reconstruct, they can reassure this study had been conducted by you ethically and scientifically. Those documents are so important. So this area is become now most integrated part, most important part. Excellent indexing because of software. Software can do automatically sorting and indexing. So it's very easy to sort the data when you have software control softwares. You have everything now in software, so you can locate very easily. You updated your system along with that physical indexing. So you can find in minutes your files. But earlier, if I used to say, okay, give me Simba's file, which is being done in 2004, and I'm asking in 2006, it used to take maybe a day. And if there is any mistake, then even a week. Use data anyways. So now, which was very difficult to locate, retrieve means removing the data. It is very easily locating, retrieving, and then replacing. Replacing is also easy. Earlier, replacing is also like if anything is happening here, replacing again, you have to go check and then place, then you put physically. Okay? Because now software will tell you first, okay, this is only the rack, this is the sub rack, here you put. You directly go and put there. So it is easy because you can see, you have even tablet in your hand. You can see, okay, this, not this B, A rack, this sub rack, and this is done. Right? Fully automated. This is fully automated. No intervention is required now. And, but here it was missing data, misplaces, everything used to happen because of manual processes. It is very rapid now. Archivist job was hectic. Very boring also. And that's why a lot of mistakes. Key resources wasted for doing and delays used to happen. A lot of time for delays, QCs, inconsistency. Training was not easy. Used to take three months to train a single guy for only archiving. Now training is very easy, less interventions. You can do, training is important, but training is easier, manageable. Key resources again for better job, not only for just doing documentation and checking, 3 checks through 2 signs, all those things not required. Inspector friendly, highly inspector friendly. Okay, so I'll show you now what is the benefit through one example. If you have laboratory information system in place, or laboratory informatics in place, application software required for the different part of the lab, what will happen without lab informatics and with lab informatics. So you remember, I think it's long time back I have explained, but if you remember, there is a flow of bioanalytical work. There is a flow of bioanalytical reporting also. So how we start? We have 3 parts, and SOPs also will be for 3 parts of any lab or regulated bioanalysis. Method development, method validation, project sample analysis, which is called bioanalysis. So these 3 parts will be done in the lab. Now everything starts if one project comes inside the lab. Okay, you need to do analysis of paracetamol. So delegation of project has to be done. Written documents, there we fill all the information, ID, YD, what kind of people, assignment of equipment, which equipment you are going to do, that will be done. Protocol training, SOPs training will be done. Method SOP training will be done. Then protocol, study protocol, clinical side, how they are doing, that will be done. The analytical part also they do incorporated in that. So that thing will be done. Then we'll go for stock weighing. We will go for spiking, addition of drug into plasma. Then we mix and store. Then we retrieve these samples. We will process based on requirement. With the batch, we retrieve standards, quality control samples, and then unknown samples, what we need to analyze. So we create an analytical batch, the whole bunch, 100 samples, 200 samples, and data generation. Then once this analysis is done, data generation will happen, compilation will be done. Then there will be a bioanalytical monitors, bioanalytical quality monitors will check this all things, all processes, all data. Report generation will happen after that, because we send the final data to the report team. Report team will generate the report, prepare the report. Then QA, everything will go to the QA. Quality assurance people will do. If they have any findings, they send to the department back, lab back, and final report. Everything will be finalized once this is closed. Gaps are closed. Final report will be released. This is what the process before LI. And then what happens? This report is prepared, QA report we say, will be sent to the sponsor. But with the introduction of LIMS, laboratory informatics, you just see after processing. So we have how many steps? Analytical batch creation, data generation, compilation, BQM, report generation. It used to take at least data generation, compilation from this time to QA finding. These 4 steps used to take at least a week. Even for a very small study, at least a week for analysis to prepare this final, to reach to the final report and to send this final report to sponsor. Used to take at least 3 to 5 days. Now, because of introduction of LIMS, these steps will be removed. Now, understand, steps removed. Analytical batch creation. Somebody is creating an analytical batch on machine. Somebody is again, machine is running, then data is generated. Compilation has to be done, data compilation, integration, compilation. Then bioanalytical monitor coming, checking, report generation is happening, report team is working, QA team is working. And then these many processes and people and time. Understanding? Final report. Now with the introduction of LIMS, everything is removed, done by the LIMS within the system. One person is doing all those jobs, maximum 2 persons. And this report is going to go to the sponsor. So without limbs and with limbs, you can save 3 to 5 days. Now understand, this is only for one project. We do at least 200 average. If somebody is having 10 LCMS, they are doing at least 200, 100 to 200, 150 to 200 projects. So how many days you are saving? So it's a huge investment initially, but then it's a huge saving and profitability, including good quality. And then you can get repeated customers. The main business happens when customer will repeat buy, repeat use. So when I talk about limbs, well-established limbs, it is well-established system in the areas, but still after limbs also there are some gaps. And that's why I'm saying there's a highly dynamic area regulated by analysis. So what is there? Data movement, now it is fully established. Process is established. Users are trained on tools like with limbs, and without limbs also people are doing managing with the software's applications. Limbs is a integrated system. So you can have one software which will integrate and do all the things. Users are trained, compliant to regulatory policies, requirements, good services, application support available. Now, when I talk about data movement is well established. Most of the labs across the globe, including India, they are compliant with the data movement. Data archiving, there also with the introduction of local or international SDMS system, we have different vendors. People adopted those based on their cost competitiveness, but still they have system and they are compliant. Highly integrated, established, users are trained, compliant to regulatory policies, good service and application support available. For these two areas, data management and data archiving is done. And because of this now, what is happening? Less documentation is required, less QC is required, quality monitoring is required, less QA is required, less manpower is required. So we are saving time, money, and so many other things, documentation time, you understand. Even tedious job, boring job. People are more interested in other things. So we are using that people, resources, time, everything for better things. But still there are some missing links like sample processing. The sample processing area, like where we treat the sample, captured manually. Still a lot of processes are being captured manually. They are ticking in forms or something. Serious manual interventions are there. Process is prone to error, inconsistency. Wherever we have manual intervention, you understand there may be challenges. There may be challenges. Non-compliance, no reconstructability, because if data entry missed due to manual processes, no way to reconstruct the processing. If something happens, like half of the ISTD or some pipetting error happened, then there is no way to, and we missed to write also. Pipetting error, we did the best pipetting, but results are showing there was some error. But we write volume done, pipetted write volume, tick. That's what we do. Now tick. Now how you get to know? There is no way. Led to serious non-compliance to GLP. GLP says results are showing error, your documentation is saying not error, so you are not following the system. GLP says what you are writing, follow. What you are following, write. If you are mismatching, no GLP. Then they may issue serious letters. And this sample preparation, because we are trained people like me, we trained, like auditors also trained, inspectors also trained, who are coming from for these audits. So this is a favorite area. They know where people are still using manual interventions. Other missing links, sample identification, shipment, storage, so sample transfer from one place to other place. Like data transfer I discussed, that is now okay with LIMS. But sample transfer, clinic to lab, lab to other place, PK site, or you have clinical different lab, different place, different countries, and there is a challenge. Barcoding, sample barcoding, people are planning to automate this area. So that is what happening, developing. Now some of the labs in India developed also, but still there is a lot of work required. Connectivity, I'm talking about today's lab, okay? A lot of labs doesn't have this gap filled. Anyways, so connectivity, compatibility, inter-lab connectivity, inter-vendor connectivity, training, application support, cross-vendor meets and relationship. These are the areas there people are lacking, like inter-vendor connectivity, like Waters, HPLC, AB SciX or Thermo MS. Now they don't have connectivity. Earlier they didn't have. Today also they have connectivity, but they have some intervention devices, but those are not fully compliant, fully supportive. Some of the features we need to sacrifice when we have two vendors. So when you have one vendor, it is good. Two vendors, but sometimes we know Shimadzu's HPLC in our area is better than Waters, and Waters MS is not okay. So we have to go for SAX. This is my experience. So we have Shimadzu, and so they have good connectivity, but so many other vendors, they don't have good connectivity. Some of the features we need to sacrifice. So there we have scope of improvement. People should, sometimes people even don't realize these are the challenges. So we have to have people who are continuously evaluating our system, existing good system, working system. We are evaluating continuously, evaluating and getting these gaps so that we'll be prepared what to be improved. About conclusion on Green Lab first part, where I say a lot of introduction of small applications and software solutions, we can make it green, almost paperless and very efficient as compared to past. So efficiency is, you can understand now. If I talk about, don't you think maybe 40 to 50%, maybe 30 to 40% efficiency improved because of only these solutions? Now, that is true actually, because it is my personal experience. 30 to 40% efficiency improved in some months only. Implementation took some time, but then once people trained, they started getting 3 to 4 days only because of single thing. You can understand. So earlier people were reluctant. They used to tell, no, it is not user friendly, sir. LIMS is very tedious. We are not IT guys. Variation in data. They say, no, when we are using it, still variation is coming. I said, it is software, it will not be. There will be some of your challenge. You put wrong, you give wrong input, that's why wrong output. Machine is machine. Machine will give you what you give the input. So they realized they made mistake, not machine. Create confusion. They say, sir, it is confusing. Earlier was simple because they were trained on that. Can't be customized. I say it is customized, but first of all, you should be clear what you need. First, you should be clear in your this thing. So we got support from engineers, software vendors. They did that. It's complicated. That was the overall. Like it is confusing, not user friendly, complicated. Earlier experience when we introduced. But once it is introduced, they learned, everybody learned, slowly everybody learned in lab. And now you say it is not only user friendly, it is actually sponsor friendly, because sponsor can take your data remotely in their computers, reports, concentration, everything, because it is in now software in your computers. They can have sharing, legal sharing through IT password and everything, and they can check your data. They can have video calls, they can do. Even our Canadian vendor supporter from Canada, they used to take our machines online, running machine, and they used to do engineering corrections from their Canada. That is what the level of support they were giving. I'm talking about machine, but this is about software. So now people are having that experience, including me. We are having more accurate data, result confusion can be customized to any level as per regulatory requirements. So it's having that adaptability feature also, keeping future in mind, and it's very straightforward. So it is very much now we are moving to the green environment. There we have almost no paper or zero paper, which is possible. So I say today we are very close to 90–95%. It is not far when we have almost no written down things in lab. Let's understand. So from where I'm coming. So now till this part, almost 75% of my lecture is completed. I talked about the dynamism, I talked about the changes, I talked about the basics first. Then once we understood the basics, then I talked about why this is dynamic. Once we understand dynamism, then I try to say what kind of solutions you can have, what kind of attitude, scientific, regulatory, quality, and other things like solutions you have in your hand. But ultimately, one most important thing which you need to have to become progressive, if somebody wants to have green lab, like exactly the most advanced lab as per today's scenario, then one thing which we need to understand is change. Change is something which will come to you. So better to change first, right? Otherwise auditor will hit you and then you change. So if we understand one philosophy, which I understood very early of my career, early stage of my career, I realized this thing. So if you want to survive, even if you want to be successful now in present and future, then we understand this theory, which is being given by Darwin long back. But this is the base of my whole growth in my career, you can say. So it's a natural selection. So what this theory says, theory of evolution. So Darwin's theory, everybody knows, I think, but I try to draw something. In short, in crux, he says, the species which is capable of adapting itself frequently for rapid environmental changes will only be survived. So you have very good example like we have. Dinosaurs were prevalent across the earth in some era. Okay, today we don't find them. What was the single most important reason they are not today? Probably they couldn't survive with the environmental changes. They couldn't survive because they couldn't change themselves as per the changes happening that time in environment. But human was there in the same era, maybe after that. And whatever changes happened up once human come in life, when human life came on earth, human even today surviving because we are ready. We have that inbuilt capability of adaptiveness. But because of our mindset and wrong notions and concepts, we start resisting because maybe our overlearning, ego, whatever we say. But naturally we are adaptive. And that is what instinct. So what this theory is, I understood something. Challenges, even today, we are having. Tomorrow we'll be having. Today we are facing COVID. You can understand. Nobody predicted this. But we are facing, we are sitting. Today we are learning. We are learning because we are surviving. So challenges are there. Now, if I talk about lab related things, analytical challenges, lab challenges, requirements, expectations, not only sponsors, not only management, I say, it is about regulatory inspectors. They have expectations, sponsors will have expectations, okay? My management will have expectations, standards will change because it's a dynamic area. It keeps changing, so we need to be as per the changes for good. Competition will be there. So you want to be innovative, you want to be different. How you are different? Quality is something which is very good. If you take quality, I want to have the best quality person, best quality lab. That is the best thing. But the worst thing with quality is also moving object. Quality, definition of quality is it's highly dynamic moving object. It will change with the time. Good quality. So earlier in the beginning you say good method is the quality. Now I say good method within the time frame you develop is a better quality. So that is what. Conditions, quality, financial scenario keep changing. Keep changing. So survival of the fittest, in short, this is what the interpretation from my side. Adaptability is the willingness to accept challenges. One must be equipped with the required advanced skills to cater the present and futuristic job requirements efficiently, possible with proper vision, strategy to grow appropriately first, and then exponentially. There is a blast in growth. If you follow things correctly, if you are quality people, nobody can stop you. You will be super, you will be number one. So while operating from India, we were number one in Asia. We were able to grab things from Pfizer and Novartis kind of big corporates in India, sitting in India. We did job for them. We did a lot of analysis of their innovative molecules. So that is what happened. So I see, even I can't imagine this kind of success we could have done, but that is possible, right? So if you do things correctly, whole world will appreciate you. It doesn't matter from where you are doing the right job. So green mint was, again, I am trying to remind you, quality, science with ethics, alert, ever ready. You should be. Gaps still are there, even after understanding the automation part. Latest system I explained to you, still there are gaps. Sample processing area, okay, right? Trainings, kappa, adapted. So ultimately fit for purpose. So let's move. What else is remaining? Probably the lab I'm showing you. This is what a lab. Earlier also I showed. There's a new building, new system. I shown the photo earlier. But what else should be there? So now, before moving, this thing I want for everyone to understand. Bill Gates, a great one. He says, change yourself before change. Otherwise, changed you. So change yourself before change. Changed you. Very important statement. So what we do when we want to create a big, good lab, green lab, we have to start from planning. We have to have proper plan. There's a floor diagram layout. There we have proper space for sitting, like this is the sample processing area, bench. We do processing by sitting. This is the lab, and this is, these are the lab benches, LCMS benches, 1, 2, 3, 4 rooms. There are 6 rooms, 6 machines, 3 machines. This is the documentation meeting room. This side, if you come, these are the sample storage, deep freezers. This is a high-end processing area, highly sensitive machine and light-sensitive drugs we can handle. This is the open common area. There we have big processing room, 10 to 12 people can work. These are the few modes. These are the light-sensitive area for this. This is light-sensitive and a separate area for high-sensitive molecules. This is wash area, washing, right? This is the balance area. Here we have a small balance room. This is the entry exit. These are the benches, side benches. Okay. So we have to plan everything in advance. Then we go for construction. Now what else? So today's advanced lab, when I say green lab, operating in today's world, where they are focusing for local support, clinical studies, and even for internationally. They can work as a standalone biomedical lab or as an end-to-end solution, along with collaboration and other things. Services for studies conducted at other sites globally also. So local and globally, they are ready to support locally, like in India, and globally also. That kind of system they should have. They basically, if I talk about biomedical lab, they are focusing on method development, but they should be very clear and equipped to have technology transfer kind of thing also. Like if somebody has already developed method, we should have enough setup machines and everything in place. Same machine, what they have developed, if we can transfer their method and we can implement faster. We can do their job quickly, give them results so that we can create value and we can get repeated business. Even then they can give the new molecules which we start from development. So first of all, we should be clear with method transfer. Those kind of services also we should be able to provide. So biomedical lab not only doing development, they are working for method transfer, technologies transfer also. Then they are working for validation. We can do developed method we can take and validate for them. Others, we can do rapid turnaround bioanalysis. We have already method in place. They just give samples, we analyze and give them back. Focus area, standard machines, we have API 3000, 4000, my people are talking about 6500. Okay, this is from AB Sykes, this is from Waters, Vivo, Vantage from Thermo. So advanced machines and sensitive machines people are having, new labs I'm talking about. Highly sensitive assays like sub-picograms, femtograms, people are talking about handling. Tissues, not only plasma, blood, tissues, patient samples, animal samples. Then specific assays are to be handled through dedicated sample prep area. As you remember, here we have this room is very high-end processing lab, high-end processing area. So highly sensitive and special molecules can be handled separated from this area where we have common analysis. Now developing, so this is done, developing assay for challenging molecules like the molecules which are already present in blood. So you will not get blank samples. We have different technology, techniques to do this kind of analysis, hormonal products, oncology, anti-cancer products, chiral assays, where MS will not work. So we need to get these drugs separated through column first, and this in time, in space. So we separate, and then we get, based on RT, you identify which kind of chiral you are getting, which kind of form you are getting basically, right? So those kind of chromatographical things, simultaneous quantitation of multiple analytes and not single analytes. People are expecting if there are 3 drugs like antiretroviral, we have combination of drugs for treatment. So all 3 drugs and their metabolites should be analyzed in single run. So one run is having 6 values. Those kind of advanced things people are expecting, and these labs should provide or having high throughput assays. In a day, 400, 500 samples. We are single machine. And then in a day, thousands of samples using 6 machines, 7 machines, 10 machines for single molecule analysis, single analysis. We are doing 2,000, 3,000, 5,000 samples, 50,000 samples in 10 days. That kind of throughputs we are talking about. So how much money, how much work, and that quality and the efforts you see, how many efforts people are working on single project, and we are doing in 5 days, 10,000 samples. How many things we are doing. So that time data integrity and all these things challenges happens if trainings are not okay, SOPs are not okay, people are not understanding what kind of expectations from them. That is all. So which kind of analytical expertise you need to have? Sample prep, you should be thorough. Chromatography, you should be thorough. You should be thorough with mass spectrometry. You should be thorough about special techniques like HPLC, ultra high performance liquid chromatography. Now UPLC, people are talking about ultra performance liquid chromatography, chiral chromatography, simultaneous quantitation. All these things we should be aware. It's a standard chart where we have, I have already explained on an earlier part, where we have the proper trained organogram for people. So organogram of people who are properly trained for their job. All these jobs, on these jobs, all people should be properly trained and evaluated. No more, no less. Okay, no more at least. No more responsibility. Auditors will check. Senior group leader can take 14. No, single not. So 5 senior group leader. So each group leader may be managing 3 group leaders. So that ratio they will check. Otherwise, they say it is impractical. So bioanalytical flow, we know sample tracking. Everything starts from here. Then we go for development, preparation of analytical standards, QC, sample preparation, then sample analysis, data processing, acquisition, everything happens. Notebook, ancillary data, we will cross-check, compile everything. Reporting is the flow. And what we use LIMS, we'll use LCMSMS as a machine, main machine, programming, automation, different places, standard guidelines, operating procedure everywhere, and information management system. So this is the ideal lab. This is the green lab I'm talking about. And these are available. And the green lab can be called as green lab, which is ready to adapt. Remember this. So project management, Western style. Sitting in India, you should understand what is project, what is communication. Communication is not only when I do good results, when I get method developed, only then I share with them. If there is a failure, I say, no, nothing happened. Today I'm not sharing with them. No, openness is required in communication. Good, bad, whatever is there, every day, if somebody is agreed, you give reporting to every day, share with them. Whether it is bad also, give them. So that they will be mentally prepared, physically prepared. Because they need to report others, they need to involve other departments for these projects. So timelines can be maintained. Timelines is everything. Today, we don't say innovative, we say fast. Today is the world of fast and first. Who comes first wins. Quality, I say quality can be innovative. Quality can be resolved and attained through innovative ways. Innovative in our own approaches, right? Consistency, honesty and integrity. Yeah, honesty and integrity will give you consistency. This is a part of culture. Sound relationship with the customer. That is what PM, project management approach. To nurture individual growth. Growth not only for our own employees, it is for our partners also. What kind of regulatory goals? I should be mentally prepared. As a lab, we should be mentally prepared to work for laws and regulations and policies of FDA, like America, Brazil, World Health Organization, everybody knows, and Europe, MHRA, this is Austria, and then All these big countries, big spaces, big origins, you say continents. So I should be ready to learn their regulations, and as per that, I should be prepared. So regulatory goals. Now I'm done with the technical part, most of the technical part. What else? Lab. When I talk about future perspective, and I say a green lab. Triple E, I intentionally have done. Green means what? Again, I'll come to the end of this presentation. I'll try to explain you. So what is happening from last decades? Now 10 years. A lot of pharma R&D. So why I'm sharing this? This is important. So if you want to sustain and you want to grow, a lab has to grow. Lab has to be green when it is sustaining and growing. And only green lab can do that. So what we need for green again. So what is happening? First of all, understand what is happening. Then we identify the points for our preparation, where I should be ready. So pharma companies and R&D going through very dynamic changes. Mergers, acquisitions, you might have heard Mylan, Pfizer merged. So many big companies used to there, they merged. Google invested in Reliance. What is this? Something big happening. Telecom, Idea, Vodafone merged. In pharma also, a lot of mergers, acquisitions happened last 10 years. Closing of sites, outsourcing, they are having now closed down their sites like bioequivalence, they are not doing themselves, so they need to outsource. Out licensing, even the molecule they are selling off, so generating money to sustain and go for development. So they do develop and sell their products. Co-development approaches are coming, risk sharing. So what ultimately the whole world is moving, they are trying to share their risk. If they themselves alone developing a molecule for 30 years or 15 years and it is failed, the whole risk come to the one shoulder. If we have co-development partners, then the risk will be half. So this model is giving lot of opportunity to third parties like contract research organizations, contract manufacturing. So things are moving, and for India it is good because we now with the in COVID situation, the kind of attitude India shown to the world, Bharat is becoming Bharat actually. So there we are going to get a lot of good work from regulated markets like US and Europe. Because of this reason, we are anticipating huge growth in coming years now for Indian pharma and other sectors. Downsizing across the globe, it is happening, which is good for now us. It happened in India also. So it was bad. But then now I'm seeing a lot of good things. Anyways, bioethical research is also on the same path of dynamic changes. Because of these things, all these things, outsourcing, merger, acquisitions, closing of sites, de-risking, risking, covalent, co-development approaches, downsizing, we are also pressurized because we are also going for mergers, acquisitions, so pressurized, existing one. Do more with fewer resources. Budget is less manpower, more pressure for more output, faster method development with the same resources, more sensitive methods with the same machines, same sensitivity machines. Difficult, but that is what efficiency innovation require. So pressure on lab for quality, efficiency, and cost effectiveness. This is what we faced. I faced myself. Bioanalysis involved from, so now what is happening? Bioanalysis earlier, if you do good, fast, and cheap, was ideal. Everybody was happy. But now, today, no. They say money, I can give you same amount, but then quality, timely, and value added. What kind of value addition you are doing? How your analysis is better than before or better than other CRO? You are better than other. So they check based on quality, timely delivery, and value added. How much value you are adding. So in today's era, I say there are 4 pillars of bioanalysis. 4 pillars of bioanalysis. For future perspective, when I talk about green lab, there are 4 pillars, I say, best of my knowledge. Somebody has to grow into that area, then understand the quality aspects, understand timely delivery is the most important aspect today, which is, I say, first and faster. Value addition has to happen. Like a bioanalytical lab, if it is operating, if it is not able to show the validation to the development chain or to the client, it is gone. Business sustainability has to have there. So it is not only operating the bioanalytical today, if something is running good, it has to show it will be there for next 25 years. So there it comes to the change, adaptability, and progressiveness, survival of the fittest. So it is a continuous process because the dynamism is the integral part of regulated bioanalytical lab. So we have to be on toes for development and growth. Creating space. Now, whatever is happening now, if I say it is dynamic, I have to be dynamic, then what is happening? A lot of creation of good jobs, good things for progressive people. Creating space for new solutions, services, and players. How? You know, automation will always be useful. Robotics, applications, software, okay? Data integrity and data management. If you want to do properly, you have to have LIMS, e-forms, electronic forms, electronic CRFs, case record files, it is in clinical side, e-notebooks, laboratory information system, scientific data management system. These applications and software solutions will be helpful for data intake, data quality, and data management. So these kind of solutions we need to introduce upfront. Value-added services like techno-commercial solutions, we need to go with techno-commercial solutions to the client, not only technical, not only commercial, based on that. So we will be more innovative in our approach. We should be continuous training. Ultimately, if something is dynamic, to cope up dynamism of anything, we need to be progressive. And for that, we need to train ourselves. We need to train. That is only the way. Training can be general or specific. So finally, takeaways of both the parts. So this is what is probably the last slide. So I say we started from everything from dynamism, understanding of analysis, and dynamism, solutions, different solutions, challenges, solutions, again, the theory of adaptability, and then finally, what we need to survive even in future. So bioanalytical science, what I believe as a takeaway, the first takeaway is an integral part of drug development process. From last 2 decades, as we discussed throughout the presentation and lecture, bioanalytical community witnessed a significant level of dynamism in bioanalysis, whether it is science, technology, quality, or regulatory aspects. So what I need to do, or we as a team need to update our tools, whether it is knowledge, technology, automation, regulation, systems, existing quality management systems, people, or culture, with the time to meet dynamism of this area. Dynamism of current scientific standard, regulatory requirements, and best quality practices. So I have to be ready for inspection. So my lab has to be inspection ready. Most important thing, if I'm passionate, if I'm doing the things correctly, I need to enjoy the job. So bioscientists, managers must understand another important area when we talk about techno-commercial aspects and innovation. 4 pillars of bioanalysis, which are quality, timely delivery, value addition, and business sustainability. We need to thought of in advance where we are going and what kind of things will be helpful to sustain and then further grow. So finally, the take-home message from this lecture can be adaptability to current disruptive challenges, which are not under our control, adequate knowledge of which are under our control, adequate knowledge of current science, technology, quality practices, and regulatory requirements facilitates bioanalytical or whoever wants to grow, bioanalytical scientist with a good insight to cope up with an extremely dynamic nature of bioanalysis, especially regulated bioanalysis, and a long sustaining lab, thus making them adapted and suitable to work in a progressive lab, which is called green lab. For a green lab, we need all these things in place. Especially, I should be mentally ready for whatever is coming on my way to cope up. to win basically. So thank you so much. And I would like to stop here. So thank you so much for your patience and enjoy the learning.
Thank you very much, sir. It was a very nice and informative session for all. Once again, thank you very much.